All right, so welcome to lecture 29. And again, every class I'm going to show the exam time so that we're all on the same page and we all know where it is. We had talked about microtubule formation, and we initially looked at it in vitro. And we learned that if the concentration of the alpha-beta tubulin dimers was above this so-called critical concentration, they would spontaneously form microtubules. So there's no additional component needed, which is pretty cool. All of the interactions are non-covalent. So this means that they can be sort of broken and reformed with low inputs of energy. And so what we end up with is a net growth at the plus end and a net loss at the minus end. And that then contributes to what we call treadmilling, where if we have a net gain of the dimers at the plus end and a net loss at the minus end, it means that sort of overall what we end up with is a migration of the microtubules from the plus to the minus end as new tubulin dimers are added at the plus end and removed at the minus end. We know that the alpha-beta dimers are bound to two molecules of GTP, and that in the beta tubulin, which is at the plus end, the GTP can be hydrolyzed. And that leads to instability of the microtubules. And in fact, if the tubulin dimers at the plus end are bound to GDP rather than GTP, we get a destabilization and a collapse of the microtubule, which we can call a catastrophe. All right? And we compared the dynamic differences between the plus end, where we see these huge gains and drops, with the minus end, where things are much more conservative and there's smaller gains and losses. When we are looking at these in cells, often the microtubules are nucleated from an MTOC, or a microtubule organizing center. And the two types of these that we talked about were centrosomes, and here's a centrosome, and here's a centrosome. And the one that I think I might have forgot to mention were these ones called basal bodies, which play an equivalent role, okay? And so they still have these gamma-Turk complexes. And what those gamma-Turk complexes do is, again, they initiate nucleation, so they sort of start the process, but they also serve to stabilize the minus end because they're bound there, okay? And this example is showing how some microtubules utilize an MTOC and other ones don't. So for example, in the dendrites, no MTOCs are involved, and we get like a random orientation of the microtubules in terms of where the plus and minus ends are. We then moved on to talk about microfilaments and actin. So these are sort of long and flexible, and they're often found under the plasma membrane, so they contribute to things like shape and motility. Again, they still have a plus and a minus end. The plus end is also called the barbed end because if we look at it, and I wish I could blow this up a little bit, we can see that they're sort of shaped a little bit like arrows. So there's a barbed end, the plus end, and the pointed end, which is the minus end. So importantly, these are regulated, often regulated by cell signals. Okay? And what the signaling complexes can do can determine which enzymes that contribute to the formation of the microfilaments, which ones are activated. And if we activate the ARP23 complex, we get cross-linked types of microfilaments because the ARP23 nucleates, but it's most efficient when it's bound to an existing microfilament. So we get this mesh-like pattern we see at the top. In contrast, foramins work at the plus end and recruit globular actin to the growing F-actin, or filamentous or fibrous actin. And so this leads to long, unbranched microfilaments. And a lot of this regulation, we have some, of course, important monomeric GTPases in the Rho family. And these include Rho, which when it's activated leads to formation of stress fibers. These are important contractile molecules in non-muscle cells. So they can lead to contraction of a cell, but they're not, it's different than what we find in muscle cells. Lamellipodia, which are kind of like two-dimensional ruffled edges that stretch out. And Filopodia, which are, we call them one-dimensional, but of course they're not truly one-dimensional, but these long, very narrow processes that come out of a cell. And I want to point out something important here, which I didn't talk about, and that's that if we were to look, say, at any of these features, including a Filopodia, so here we would have this small cluster. Oh, PowerPoint. So annoying. So they form these long filaments. What's important to note is that these are still cytoskeletal. They're still within the membrane, so it means the membrane gets stretched out over these long, narrow sort of strands. So these aren't like cilia or flagella that you may see in other courses, where those are actually external protein features. They exist outside of the plasma membrane. These are actually still under the plasma membrane, fantastically enough. And so today what we're going to look at is cell-cell junctions. And this is going to start tying in to things we talked about really early in the course. We're going to look at cell extracellular matrix interactions as well, and the importance of something called integrin signaling. Okay, and finally, we're going to introduce our last topic, which is cancer, and we're going to talk a little bit about epidemiology in terms of that. So we can see different types of arrangements of cells. And so we might get them arranged into, say, a tissue, as we see at the top, where they're highly organized and the cells are directly connected with their neighbors. In contrast, if we look at things like connective tissues, the cells may be existing sort of on their own. They don't have direct contacts with their neighboring cells. In both of these cases, the cells do interact directly with the extracellular matrix. So they are bound to their environment, okay? And so in this case, these cells are polar, and it's the ones shown at the bottom here are not necessarily polar unless this was, say, a crawling cell that had a leading and a lagging edge. And we looked at those in our last lecture. So these are the different types of interactions that we're going to be looking at. And one of them is missing here, but it doesn't really matter. We're going to look at tight junctions, adherens junctions, gap junctions, desmosomes, hemidesmosomes. This is looking at epithelial tissue, and we have this basal lamina below that will be interacting with the cells, and then below that we have the connective tissues. And so it's highly organized. And this is like a list showing us the different type of cell-cell junctions. So this is maybe a good study aid. We're going to be looking at all of these different interactions, okay? So the first one we're going to look at are called adherens junctions. And so adherens junctions are mediated by proteins called cadherins. And these were initially identified using antibody studies, and the antibodies that would bind to the cadherins would interrupt this interaction between the cadherins, and it would cause the cells to lose their interaction with each other, so break apart. The cadherins then, if we look here, this blue area is the outside of the cell, or really the region between the two cells, and the orange is showing the cytosol of the right and left cells. So the cadherin itself, shown in blue, is actually a transmembrane protein. And within the cytosol, it interacts indirectly with, in this case for an adherens junctions, with actin. Okay? And so we can see there's a number of sort of adapter molecules, and then it's interacting with the actin microfilaments. Importantly, these catenins are signaling molecules. So this actually mediates signaling pathways within the cell. And we're not going to look at the cadherin pathways in this class, but you may see them in other courses. The part that's extending outside of the cell has a number of repeated regions. So this means the sequence of the amino acids repeated several times within the polypeptide. And these repeats interact with each other externally. There's different types of cadherin. E here stands for epithelial, and in this example, we're looking at epithelial cadherin. But the way that other, like placental cadherin, for example, works, it's the same. It's just on different tissues. So we have an indirect association with the cadherins and the catenins, and this forms or can form sort of a band around the cells between the neighboring cells. And that can contribute to the polarity of these cells. What we're looking at here is the nucleus is labeled in blue, okay, and the cadherin is stained in green. And we can see that this would be maybe looking down from the top of a cells or a tissue. We can see that there is a band of cadherin sort of all the way around the cells, okay, sort of forming a seal between those cells. One of the things that happens in cancer to epithelial cells, if that's where the cancer is they can undergo an epithelial to mesenchymal transition. And you'll sometimes see this EMT described when you're hearing about cancers. What happens is that the cadherins break down, and we go from this organized tissue to a loosely associated set of cells. And this can allow cancers to metastasize. And so when cancers metastasize, it means they move from the primary site of the cancer to new locations. And this is when cancers become really dangerous and they spread throughout the body. And so that's preceded, if it's an epithelial sourced cancer, by this EMT. Desmosomes, these serve as what they sometimes call spot welds. So instead of being a band, they're localized to a smaller location. They're mediated by desmosomal cadherin, which again functions in exactly the same way. A transmembrane protein, okay, with these repeats in the extracellular region that interact with each other. However, here, a different type of catenin called desmoplakin allows them to interact indirectly with intermediate filaments here. And so the intermediate filaments, remember, give strength. And so these sort of desmosomes provide like as they call it, these spot welds, because it sort of holds two cells together very firmly via these intermediate filaments. Tight junctions are the last ones here, and these are the last ones that we're going to talk about in terms of the contributing to polarity. And these, again, form a band, or can form a band around the cells. And the proteins here, the textbook doesn't show them in detail, but instead of cadherins, you would have occludins on neighboring cells that interact with each other, or another group which are called claudins. So claudins on one cell with a claudin on another, occludin on one cell with occludin on the other. And these occludins or claudins are also associated with the cytoskeleton via a group of proteins called ZO proteins for zonal occlusion. What this really does is it really reinforces that bond between the cells. And so what we're seeing now, and what this arrow is indicating, is that now it becomes very hard for materials, even very small molecules, to pass between the two cells because they're sealed up with these tight junctions that are a band all the way around. And so this means that for cells that have these tight junctions around them, if something wants to pass through, they actually have to move through the cell. And so this is what we see, for example, in the gut. For things to get from the gut lumen beyond into the blood, they, in most cases, have to actually move through the cells themselves, which means they can be selectively moved through. Gap junctions, in contrast, what these are allowing is they're allowing two neighboring cells to share material with each other. So the previous ones, the cadherins and the tight junctions, they're mediating or actually physically holding the cells together. Here, this is more a sharing of cytosolic components. They're made up of clusters of a protein called connexin, and there's usually 6 of these connexins in the cluster. And what they do, this is sort of slicing through one of them here, is they form a pore. And this pore is a direct link between the two cells. So this hydrophilic channel, but it's very small, 1.5 nanometers. So this means things like proteins or macromolecules can't fit through, but small molecules and ions can move through. Okay, so like a simple sugar, like a monosaccharide could move through, but not something larger, for example. And so they allow for molecules up to a molecular weight of about 1,200 grams per mole through. Questions so far? Okay. So the extracellular matrix is composed of 3 components. Okay. It's composed of a structural fiber. Okay, so this is a protein, and we're just going to focus on animals here. So for animals, this is a protein component, usually collagens or elastins. There's then this, they're calling it here components of a hydrated matrix. These are polysaccharides attached to protein. So polysaccharides on proteins. And these form what are called proteoglycans. We talked about glycoproteins, and if we look at a glycoprotein, it's mostly protein with a smattering of carbohydrate. In contrast, if we look at a glycoprotein, proteoglycan, it's mostly sugar. So mostly the carbohydrate component with a much smaller component of protein. And so those are proteoglycans, and these are huge molecules. Okay? And then there are going to be adhesive molecules, which are just flat out glycoproteins. And these include fibronectins and laminins. And so these 3 components make up the extracellular matrix. If we look at something like bone, the extracellular matrix is very dense. It's a solid, right? So it's a solid. If we were to look at something like a cartilage, it's much more stretchy, so cartilaginous. It's sort of spongy a little bit. You can imagine eating a chicken wing or something, chew into the cartilage. And then in contrast, connective tissue is kind of like jelly-like. And these serve different roles within the body and different important roles. But we can see here it allows for different types of organization. And that'll utilize different components in the ECM, but there'll still be a protein, polysaccharide, and glycoprotein component. Here's my great joke where this is the extracellular matrix. So there we go. And this is topical too because there's a new Matrix movie coming out. Unlike most of my jokes, which are all based on movies from 20 or 30 years ago. So there we go. I feel like I'm really back in the loop here. All right. So this molecule here is laminin. And laminin is a glycoprotein, and it's an adhesive glycoprotein. So it's actually composed of 3 polypeptides that are non-covalently interacting with each other, and they form this structure. And what we can see on the right here is here we have our type 4 collagen. So this is the protein component. This perlecan, this is the proteoglycan, or the big polysaccharide. And then here we can see the laminin. And what the laminin does is the laminin mediates the interaction between the extracellular matrix. So it has components which bind to, up here, this binds to the extracellular matrix. And we can see it interacting here with, it looks like the collagen. And then it has, at the bottom, this thing called an integrin binding domain, right? And so the basal lamina is what we find under epithelial tissues. And so the basal lamina has, will have this type 4 collagen as the protein under it. Okay? And they've sort of flipped it here, so the inside of the cell is on the bottom. Often when we look at the basal lamina, we have it the other way around. So just a note to keep, what we're looking at the top is the outside of the cell. It's important to note that. And so the importance here is this interaction, this interaction with the integrins. Way back when we were talking about growing cells in culture, mammalian cells, I said often we treat the plastic with components of the extracellular matrix, and I said that the molecule we often coat them with is called fibronectin. Fibronectin, remember, is another one of these adhesive proteins, similar to laminin. And what it can do is that fibronectin, which is stuck to the surface of the plastic plate, binds to the integrins in the cells, all right? And we're going to see the importance of the integrin signaling now. So integrins are made up of an alpha and a beta subunit. And it's a transmembrane protein, okay? Interestingly, the alpha subunit undergoes a cleavage, and then it binds itself back to itself covalently using a disulfide bond. That's not something I'm going to quiz you on or anything, but it's kind of a neat way that it becomes activated. This is how it becomes activated. They can work combinatorially. I think there are 9 different alpha subunits and 24 beta. So this allows them to interact with different components outside and inside the cell. Okay. They're linked, again, indirectly to the cytoskeleton. So that means there are going to be proteins between the cytoplasmic tail and the whatever the cytoskeletal element it's bound to. And there's 2 things we have here, focal adhesions. And remember, these are often associated with those stress fibers we looked at in the cytoskeleton. And hemidesmosomes, which are bound to the intermediate, indirectly to the intermediate filament. So here we can see a focal adhesion. So we have the integrins, the alpha-beta integrins, and they are interacting indirectly via these 2 proteins to actin. This is a micrograph of a cell where the actin is stained in red and the vinculin is in blue. And we can see it localizes to the plasma membrane, the vinculin, and it also localizes to the actin. So we can call this like a co-localization type of experiment where we're using 2 different fluorescent stains, saying 2 things are in the same space, so they're likely interacting. So the fact that the vinculin and the tips of the microfilaments show up in the same spot likely means they're interacting. So I'll write that down. So we call those co-localization. These are done a lot in cell biology. The problem with these experiments is it doesn't prove that the two things are interacting. It just suggests that they may be interacting. Here we can see hemidesmosomes. So again, an indirect interaction between the integrins, shown in green and blue here, through these adapter proteins to the intermediate filaments. Cool image of those here, right? Using this transmission electron micrograph here, you can really see it's just quite a dramatic representation of what we see on the left. So one of the reasons we care so much about integrins is that turns out they're important signaling molecules. And they undergo both inside out and outside in signaling. So what this means is they can respond to signals that are outside of the cell and transmit them in, or they can take a signal from inside the cell and transmit it out, oddly enough. And so when we look at inside-out signaling, what happens is internal changes lead to effects such as clustering of the integrins on the surface. So they respond to changes in the internal environment to cluster together. Okay? So that might be important for, say, the formation of a hemidesmosome where we see that integrins are clustered. In contrast, the outside-in signaling, what happens is when the integrins are bound to fibronectin or laminin, they will signal into the cell. And these will activate different signaling pathways, but these contribute to the survival of the cell, among other things. Because we talked about in our cell culture section how when they lose anchorage, the cells die, right? That's because of the integrin signaling pathways. So months and months, years ago, I told you this important fact that cells were anchorage dependent. And now, years in the future, we learn why. It's through this integrin signaling pathway. And this is just talking about a couple of the different kinases that can be activated, for example. But there's a large number of signaling pathways that are mediated by this outside-in signaling pathway through integrins. Okay, that's it. The cytoskeleton, done. Any questions? Okay, so we are going to move on to our last section here. So cancer is a good way to wrap up an introductory cell biology course. First of all, it's something that we all sort of have a stake in. Unfortunately, all of us will have some brush with cancer, either ourselves or a relative or friend at some point, because these are highly prevalent in our society. Well, I shouldn't say they're highly prevalent. I should say that, well, I'll show some slides. These do occur fairly regularly. Fortunately, most of the cancers that we see are going to be treatable, okay? And so not all are, but we've made a lot of progress when it comes to understanding cancers, and especially for treatments of cancers. So the prognosis that we give, that people get now, are much better than they were, say, 20 years ago, okay? But the other thing about cancers, which makes them very interesting, is they tie in a huge number of components of our cell biology course, and they combine them. So it's a nice way to sort of bring everything into view at the end of the course. Okay? And I guess I don't even have it defined here, but I should probably define it as what we have is you have unregulated growth of cells. And it's extremely important that cellular growth is regulated, but that's lost in cancer cells. And they have a number of other important hallmarks that we'll get to next class or the one after. So they are, but these are our cells. So this raises issues and challenges when we're going to treating them, because how do we kill a cancer cell without killing all of our cells, right? Because these are actually derived from our cells gone rogue. So today we're just going to talk a little bit about the epidemiology And with epidemiology, what you're looking at is you're basically studying the numbers of a disease. So you look at shared factors between people that have the disease. You may look at things like geography of the disease, where it's occurring. You may do things like genetic studies. There's a large number of different ways we can do epidemiology. Okay? But what you're actually looking at with epidemiology is you're just looking at sort of the numbers. You're actually not looking at mechanism or anything like that. You're saying, we had 25 people who worked in this factory, 25 out of 100 of them became very sick, got cancer. So that would suggest that something in that factory is causing cancer. It doesn't tell us that that's what's happening. That's the difference of causation versus correlation. So correlation means two things are related but don't cause each other. The great example there is if your house burns to the ground, there's a much higher chance that firefighters visited your house that week. The correlation is firefighters burn your house down, right? But that's not the cause. But if you were to correlate the two, firefighters are there, my house burned down. So that's the danger of epidemiology, all right? Is that random events do look like, so if you were to take a random sampling of locations where people get cancers, they would cluster randomly, oddly enough, and look important. So that's some of the dangers of epidemiology. But it can give us lots of information, and we're dealing with epidemiology all the time right now because of the COVID pandemic, right? So we're tracing where cases occur. How old are people? Are they vaccinated? Are they wearing masks? Are they in the same class at school? Whatever it might be. So that's epidemiology. And then the last things we're going to look at are the principles underlying how cancers develop. So we'll look at this in the next class. And then later we'll look at the importance of the cell cycle, which regulates when a cell replicates, and apoptosis. So this is a little bit out of date because it's pre-pandemic. And the pandemic will certainly affect these somewhat, these numbers, okay? It's killed, I think, something like 5 million people in the past 2 years. So that will have some effects. But if we were to take developed countries, okay, and I don't love the terms developed and developing, but that's what they're using in this slide, so that's what we'll stick with. So this would be, for example, North America, and we break down how people die, okay? What we'll see is most people will die here of cancers and circulatory diseases, okay? But what we'll see is like a pretty low percentage in a normal year. Only about 1% of people die of infections. So infections can be bacterial, viral. You're dying because some organism or virus kills you, okay? And the numbers between cancer and circulatory are, if you go from year to year, they're sort of shifting between each other. And circulatory and cancers are things that kill people that are older. So they take a long time to develop. And so this is often, we could correlate this with populations where people die much later. In contrast, infections tend to kill children. And so if we look at what we would call the developing world, amazingly, almost half of the people that die in the developing world are due to one form of infection or another. There's a huge number of reasons for this. A lot of them are kind of infrastructure based. So there might not be proper sanitation. A huge problem in the world is people don't have access to toilets. And so human waste spreads into water supplies, for example. So this is a huge problem. There may not be access to good medical care or the drugs that are required for treatment. So if the drugs are expensive or they're needed by the so-called developed world, we tend to hog them. And we've seen this for sure with vaccines, where we're on well, heading for vaccine number 3 for some of us, and other places, they're still lining up for their first vaccine. So this is an issue. But if we were to actually look at leading causes of death, and this is taken from 1900 in the USA, what you see is it looks almost exactly like what we see in the developing world. It's basically almost a mirror image there. Okay? And so this just tells us that when people don't die of infections, they're going to die of something else. And that's the real bad news here, is it's going to happen. If you add it all up, it comes out to 100%. So something's going to get someone anyway. And we're fortunate enough that our population tends to die off of things like cancers and circulatory diseases. Okay? So if we look at cancers then, we can also look at this sort of epidemiology. And what we have on the left is the prevalence of cancers. Of people that do develop cancers, the prevalence. And way and by far, the most prevalent are skin cancers, right? And followed by prostate, breast, and lung are sort of the next biggies. In contrast, if we look at people that actually die, which fortunately is a small percentage of the people that actually get cancer, and as I said, as treatments improve, less and less people are dying of these. What we see is that the really dangerous ones now, we can see things like lung, colorectal, breast, but look at pancreas here. So pancreas there's very few cases, but overall of fatalities, it represents a significant number. And so what this means is that different cancers have different survival rates. Now, we need to keep in mind that every cancer is unique and different, okay? So 2 people that have lung cancer, they actually have different lung cancer, okay? It's just cancer of the same tissues. And so there's variability here. And often we go by these 5-year survival rates. So if people survive 5 years beyond when the cancer was first diagnosed and treated, they consider the cancer to be in remission. And many of you will have heard this phrase before. So if we look at something like non-myeloma skin cancer, the survival rate is almost 100%. So we can treat these very easily. Other things like prostate, testes, breast cancer, now the treatment rates are really good. And so these are ones that are often highly treatable. In contrast, if we get down here to esophageal, liver, pancreatic, the 5-year survival rate is low. It's not 0, okay, but it's much lower. And so different cancers we can categorize by the severity. It's as I said, it's a disease of the aging. So when we look at a population where people are living to their late 70s, the likelihood of them getting cancer increases. And this, we'll look at the mechanism for that in the next lecture. But what this on the left is looking at is just age-specific cancer incident rates. So we see as people get progressively older, the rates of cancer, and I think this is per 100,000 people, they rise. They start to drop off a little bit at the end. And this could be perhaps due to the fact that at a certain point, people again become very susceptible to infection, which again, we saw that certainly during the pandemic, is that in the early stages at least, it was, at least in Canada, most of the fatalities were in nursing homes. Here we can see the Canadian mortality, and we can see we're separating here cancer, male-female, circulatory male-female. The actual numbers aren't hugely important, but what we can see is for both circulatory and cancers, they develop much later in life. So a disease of older populations. So we're still just looking at epidemiology here. Yes?
I was wondering in the last slide, it seems like the female for circulatory is like way lower. what causes a huge jump from, it looks like 70 to 80?
Oh, so what causes the huge jump? I don't know specifically in this case. No, I don't know what the big jump is. For cancers, we get accumulation of damage, and that causes the jump. I don't know why specifically there is that huge jump. It's a good question, though. Yeah, for sure. Oh, I guess one thing that yeah, no. Yeah, I'd have to think about that. I never thought of that before. Sorry. You can see my brain's working really well on the fly today. Okay, so what we're looking at here is the x-axis is age-adjusted cancer rate per 100,000 population. And these are just bar graphs looking at different types of cancers. And each one of these bar graphs is looking at a different place. So, for example, if we look at things like stomach cancer, they have a much higher prevalence in Japan compared to the US. Whereas if we were to look at, say, breast cancer or colorectal cancer, the rates are much higher in the US than we would see in Japan. And so what this tells us is that there is a geographical component to cancer. And that can be sort of twofold, right? So there's two things that the geography that could be different among the people. So what's one of them? Maybe there's even more than two. Sorry?
Culture.
Culture, yeah. So there could be cultural differences. These could be things like diet, but they could also be things like smoking rates, drinking rates, things like that can affect it. What's another one that we might have? The actual environment. So, are we somewhere that's contaminated with soil, things like that? The water, is the water safe to drink? There was one back there, yeah. Climate, yeah. So again, what's the environment? These can contribute. You got another one? Yeah. Access to medication. So this is looking more at development of cancer, but certainly if we looked at survival or things like that, it would be much different. But this one actually, I should have been more clear about that. This is just an incidence rate. There's another one, differences that we see between different places. Yeah, so there could be genetic differences too, right? So depending on the homogeneity of the population, where some places actually have a much more homogeneous population, we may see differences there that are cancers that are rare in one location are higher because if it's a genetic link, you might have a higher percentage of the population. So let's look and see what happens here. So this is looking at the environmental aspect. So what we're looking at here is we're starting out with a male Japanese person living in Japan, okay, and we are looking at the prevalence of stomach cancer versus colon cancer. And we can see that the prevalence of colon cancer is quite low. If that male Japanese person immigrates to California, the chances of them getting stomach cancer decrease drastically, but colon cancer increases. So this is then indicative of an environmental difference rather than genetic, because if I move from Japan to California, my genetics remain the same, but my environment has changed. Now, whether that's physically in the environment or some cultural difference, maybe now he's moved from a diet of something healthy to Twinkies or something. Sorry if there's anyone that's a Twinkitarian and only eats Twinkies in here. If we look at the sons of male Japanese immigrants to California, so the generation down, again, stomach cancer low, colon cancer high, and then I don't know what a native Caucasian is, but apparently that's a term they're using. Native Caucasian males in California, we can see that the rates of the sons of male Japanese immigrants are actually quite similar. So in one generation, we've sort of evened this out. So this indicates that there's definitely an environmental aspect or cultural aspect to it. I'm just going to go, this slide is showing the same thing. This is showing migration patterns. And if the arrow gets smaller, it means that type of disease, if someone immigrates, they're less likely to get that. And if the arrow gets bigger, it means they're more likely to get that. So for example, moving from Japan to Hawaii, the chances of stomach cancer decrease, but of prostate, colon, and breast cancer increase. Okay. And so this does indicate that there is an environmental factor. I think I'll save this for the next class, but there is also a genetic component. And we'll look at that in the next class. So we'll see everybody on Friday.